All the living organisms on this earth have limited lifespan. Lifespan is the time period from birth to the natural death of an organism. Lifespan differs from one organism to another. For example, an elephant is having the lifespan of about 60 to 70 years. A cow may live up to 18 to 22 years. A tortoise having the lifespan 80 to 120 years. A crocodile may live up to 70 to 100 years. While the butterflies are having shorter lifespan of about 8 to 12 months. In case of plants, rose plant can live up to 5 to 7 years. A banyan tree may have the lifespan of about 2000 years. A mango tree with 50 years of lifespan. Lifespan of an organism is not correlated with their size or any other aspects. For example, size of the crow and parrot is almost same, but there is greater difference between their lifespan. Lifespan of the crow is about 15 to 20 years, but that of the parrot is about 150 years. But whatever may be the lifespan, death is certain for every living organism. Therefore, to continue its race generation after generation, all the living organisms should reproduce before its death. Reproduction is the process that ensures continuity of life on the earth. It can be defined as biological process in which an organism gives birth to the young one of its own kind. Depending on the organism's habitat, its complexity, its physiology, and several other factors, the organism can be reproduced by two different methods like asexual reproduction, which is commonly observed in lower organisms, and sexual reproduction, which is commonly observed in higher organisms where sexual dimorphism is exhibited. Let us see some of the major differences between sexual and asexual reproduction. Asexual reproduction is often referred to as uniparental because only one organism is involved in the reproduction process and it can produce offspring, while sexual reproduction is termed as biparental because to produce the offspring, two organisms are required which must be belonging to opposite sex. Asexual reproduction involves only mitotic cell division, whereas sexual reproduction includes both mitosis as well as meiosis during gametogenesis. In asexual reproduction, there is no formation and fusion of gametes. In case of sexual reproduction, the organism produces haploid male and female gamete which will fuse together during a specialized process called fertilization, which results in the formation of a diploid structure called zygote. Zygote will further develop into the young one. In asexual reproduction, the organisms produced are similar to one another, hence they are often referred to as clones, while sexual reproduction produces the offspring with greater variation. In asexual reproduction, there is no variation, therefore no evolution process, while in case of sexual reproduction, greater variation occurs, which will act as raw material for evolution. Asexual reproduction is a rapid process, while sexual reproduction is a slow process. Let us learn about some of the different methods of asexual reproduction observed in lower organisms. Some common methods of asexual reproduction includes fission, sporulation, zoospore formation, conidia formation, budding, gemule formation, fragmentation, and vegetative propagation. Fission. It is the process in which single parental cell divides or split into two or more daughter cells which are identical to one another. If two daughter cells are formed by the splitting of single parental cell, then it is called binary fission. For example, bacteria, paramecium, euglena, amoeba carry out binary fission. If more than two daughter cells are formed by the single mother cell, then it is called multiple fission, which can be observed in Plasmodium. On the basis of plane of cell division, binary fission may be irregular, as in case of amoeba, or longitudinal, as in case of euglena, or transverse, as in case of paramecium. As you are observing here, in paramecium, there is division of both micro and macronucleus, followed by separation of two cells in transverse plane. In euglena, the nucleus and cytoplasm divides first, which is followed by separation of two cells in a longitudinal plane. During multiple fission, a single mother cell split into more than two daughter cells. Amoeba is a fascinating organism which carry out both multiple fission as well as binary fission during favorable environmental condition, while under unfavorable environment it undergo encystment. Sporulation. In this process, the parental organism produce specialized cells called spores. During unfavorable condition, within a sac-like structure called sporangium, spores are produced. By the dehiscence of wall of the sporangia, spores are released into the air. Upon getting the favorable environment, spore germinates to produce new organism. It can be observed in bacteria, algae, fungi, and in some protozoans. Here is the microscopic view of sporulation in fungi. Zoospore formation. Zoospores are the flagellated motile spores produced inside a sac-like structure called zoosporangia. Upon getting the favorable condition and suitable substratum, it develops into new organism. It can be observed in aquatic algae like Chlamydomonas ulothrix. Conidia formation. Conidium is the exogenous, non-motile outgrowth born on the tip of a special hyphae called conidiopore. It is produced during unfavorable environmental condition. Upon getting the favorable environment and suitable substratum, the conidia germinates to produce new organism. It can be observed in fungal organisms like Aspergillus and Penicillium. Budding. In this process, the parental organism produces a small outgrowth on its surface. The bud gets separated from the parent and matures into new organism. It can be observed in yeast, hydra, sponges, etc. In yeast cells, the parental organism forms a small outgrowth called bud. It is followed by the division of nucleus and cytoplasm. Both nucleus and cytoplasm move into the bud. The bud detaches from the parental cell when it becomes able to lead the independent life. Here is the microscopic view of budding yeast. In hydra, the parental organism forms a small outgrowth followed by the development of mouth and tentacles. After that, bud detaches from the parental organism and become independent organism. Gemule formation. It is the process of formation of internal bud inside the body of the parental organism. The gemule is enclosed within a chitinous envelope or in a shell with spicules. The envelope or the covering has an opening called micropyle. With the envelope, a specialized cells called archaeocytes undergo division to form the gemmules, which can be released out through the micropyle. During unfavorable environmental condition, gemmules are released out. Upon getting the favorable environment, it develops into new individual. It can be seen in freshwater sponges like Spongilla. Fragmentation. In this process, the parental body divides into two or more pieces. These pieces can develop into complete organism. In multicellular animals, fragmentation is followed by regeneration process, in which the lost body part are grown to form new organism. It can be seen in fungi, planaria, protozoans, and echinoderms. Asexual reproduction in plants is often referred to as vegetative propagation. Asexual reproduction in plants occur by different somatic parts of the plant other than the seed. These structures are called vegetative propagules. Formation of this structure do not require two parents, hence it is considered as asexual reproduction. Vegetative propagation in plants can be classified into two types. It may be natural or it may be artificial. Natural vegetative propagation may occur by some somatic structures like root, stem, leaves, bulbils. If stem is involved in the vegetative propagation, it may be underground stem, subaerial stem, or aerial stem. Artificial vegetative propagation is utilized by the horticulturist to get the improved varieties of plants. It can be done by different methods like cutting, layering, grafting, and micropropagation. Let us learn the natural vegetative propagation by different somatic structures of the plants. Some of the vegetative propagules involved in asexual reproduction includes the roots. Plants like sweet potato and dahlia have modified storage roots, which bears the bud called slips. These buds can produce leafy shoot and root upon getting the favorable environment. Vegetative propagation by stem. Vegetatively propagating stem may be either underground or subaerial or aerial stem. The modified underground stem reproduce by the formation of buds. These buds get detached from the parental body and grow into new plant. Some of the modified underground stem includes rhizome as in case of ginger, tuber as in case of potato, bulbs in onion, corm in colocasia. The modified subaerial stem reproduce by the production of new shoots or crowns. Some of the modified subaerial stem includes runner in grass, sucker in banana, stolon in colocasia, offset in pistia. The aerial stem reproduce when it is detached from the parental plant after getting favorable environment. Some of the aerial stem includes phylloclade, which is observed in Opuntia. Aerial stem of rose, sugarcane, hibiscus, etc. are the good example for vegetative propagules. Vegetative propagation by leaves. In some plants like bryophyllum, reproduction occurs by adventitious buds present at the leaf margin. If these buds gets favorable environment, they develop into new plant. Vegetative propagation by bulbils. Bulbil is the fleshy bud present in some plants like agave, pineapple, etc. Bulbil, if gets suitable environmental condition, then it can grow into new plant. Some of the vegetative propagules can reproduce at very high rate so that the plant can spread in the area very quickly. For example, offsets present in Eichhornia reproduces vigorously and spread all over the water body within a short period of time, and it is very difficult to control them, hence often called terror of Bengal. Now let us look about some of the artificial methods of vegetative propagation. It includes the first method called layering, grafting, and micropropagation. Layering. Layering has evolved as a common method of vegetative propagation of numerous species in natural environment. It can be done by two methods such as air layering and ground layering. Air layering is useful method of producing roots on the stem. In this method, a small portion of bark is removed from the stem or a wound is made. Then The wound is enclosed by moist soil with rooting medium. After some time, one can observe the development of root in the wounded area, which can be used to propagate new plant. In ground layering method, the stem is bent down and the target region is buried in the soil. This buried portion develops the root. This portion can be separated from the parental plant and can be grown independently. Grafting. It is the technique that joins the two plants into one. In grafting, the upper part of one plant is grown on the root system of another plant. The upper part is called scion and the rooted part is called stalk. It is the most common method used in horticultural technique, whereby tissues of plants can be joined so as to continue their growth together. In angiosperms, grafting is possible only in dicots but not in monocots, because monocot plant lacks cambium, which is needed to form secondary xylem and secondary phloem. In grafting, it is very important that the plant should form primary and secondary tissue for its survival. Micropropagation. Growing the plants in a large scale through plant tissue culture under in vitro conditions of high light intensity, controlled temperature, and a defined nutrient medium is called micropropagation. So to conclude, asexual reproduction involves a single parent and results in offspring that are genetically identical to each other and to the parental organism. In asexual reproduction, the population of an organism can increase rapidly. There is no requirement of sexual partner. This is the most common method of reproduction in single-celled organisms. All the prokaryotes reproduce asexually without formation and fusion of gametes.